Good morning. Welcome back to Open Med School. Today I am going to discuss with you about resting membrane potential, which is a very important concept as far as excitable tissues are concerned, especially in nervous system, cardiac tissues, musculoskeletal tissues and many glands also. If you use a microelectrode as shown in this picture, the recording electrode is kept inside the cell and the non-recording electrode outside. There is a potential difference between outside and inside. Inside being negative compared to outside. Inside, so it is mentioned as minus 70 to minus 90, meaning that the inside is more negative compared to outside. This is called the membrane potential. In the resting tissue, there is a potential and that is called the resting membrane potential. Hope this concept is clear to you. Now let us think why or how this is maintained. So take a cell. So this is the cell. As mentioned, the inside of the cell is negative and outside is positive. And when you record it, this will be around minus 70 to minus 90. So this is the resting membrane potential. We will take a view of this cell membrane. The cell membrane is semi-permeable. That means some ions are only freely permeable, some others are not freely permeable. They need some transport systems. That is why it is called a semi-permeable membrane. So let us take the inside and outside of the cell. So this is the inside and the other portion is the outside. This is the outside. The major ions are charged particles. For example, sodium is positively charged, potassium is positively charged, chloride is negatively charged and protein is negatively charged. These are ions. So ions are charged particles. So take sodium. Inside the cell, sodium is only around 15 milliequivalents. Outside it is high, it is around 145 milliequivalents. So inside sodium is low, potassium is high. Take potassium. Potassium is in the reverse. That is, inside is around 155 milliequivalents and outside is only 5 milliequivalents. So it's a very important point to be remembered. Inside potassium is high, outside sodium is high. Take chloride. Chloride goes along with sodium. So what happens? Outside it is around 120 milliequivalents, inside around 4 to 5 milliequivalents. Then comes the other anions. So main anions are phosphates and proteins. So it consisted around inside the cell around 100. So this is there is a concentration gradient between inside and outside and there is a semi-permeable membrane and some of the ions will diffuse, some needs active pumping or some channels are required. Let us look into this matter little more detail. 40% of the energy of the cell is for the sodium potassium pumping. 3 sodium is exported for 2 potassium inside. So it is called 3 sodium 2 potassium pump. So here there is a channel, so sodium potassium pumping is going on. It requires ATP. ATP is converted to ADP and this energy is used to pump out sodium and pump in potassium. 3 sodium 2 potassium. suppose the cell was having, there is no potential difference between inside and outside, take it as 0. By sodium pumping, what is happening? The potassium is getting inside and more sodium is going outside. So, inside the cell becomes negative. This approximately constitutes around 5 millivolt. And now you know that the potassium is high and the potassium will be passing through leaky channels. So, there are free channels through which potassium can pass on against the concentration gradient. So, here you know that the potassium is high inside the cell. So, the potassium will leak outside. And remember, potassium is attached with anions, negatively charged. They are mainly the phosphates and the proteins. So, once the potassium is going outside, what will happen? The cell becomes more negative. So, this negativity increases to around minus 90 by the transfer of potassium. That is, this produces an potential difference, and this approximately constitute around the potential, resting membrane potential comes to around 90. Then there are leaky sodium channels. So, what happens through the sodium channel, outside the sodium is high, inside the sodium is less. So, the sodium will move inside. Sodium is positively charged. So, once the sodium enters, what happens? The negativity decreases. So, during rest, the membrane potential is somewhere around minus 70. This is in the case of a nervous tissue. In different tissues, the resting membrane potential is different. Hope this point is clear. So, we can say that the resting membrane potential is maintained by, one is the sodium potassium pumping using ATP, second is the leaky potassium channels which contributes the major part, and then the leaky sodium channels. So, interplay of this leads on to or maintains the potential difference between inside and outside cell. We have only taken 2 cells, 2 ions. So, remember that there are other channels also. There are calcium channels, chloride channels. All these can contribute to finer changes in the membrane potential. So, membrane potential or the resting membrane potential is the potential difference between outside and inside when recording with a microelectrode. The inside is negative, and we have studied the mechanisms of maintenance of the negative membrane potential. Hope this is clear to you. We will come with the Nernst equation and equilibrium potential in the next session.